Assalamu alaikum wa rahmatullahi wa barakatuh. MCQ for the laboratory scientist and technologist license exam in microbiology. First question: proper media for culture of a urethral discharge from a man include sheep blood agar and phenyl ethyl alcohol agars. B. Eosine methylene blue and sheep blood agars. Thioglycolate pros and chocolate agar. Chocolate and modified tire Martin agar. The correct answer is D. D is the correct answer. Chocolate agar and modified tire Martin agars should be used for recovery of Neisseria gonorrhoeae from urethral discharge. Chocolate agar provides the nutrient required by Neisseria gonorrhoeae, and selective media contains antimicrobial agents that inhibit other organisms and permits recovery of pathogenic Neisseria. So, correct answer is D. Question number 2. A sheep blood agar plate inoculated with 0.001 ml of urine grows 70 colonies of Staphylococcus aureus. How many colony forming unit per ml of urine should be reported? A 70, B 700, C 7,000, D 70,000. When 0.001 ml of urine is plated, the growth of one colony is equivalent to 1,000 colony forming unit per ml. Thus, 70 colonies is equal to 70,000 colony forming unit per ml. So the correct answer is D. Third question: the lowest concentration of antibiotics that inhibits growth of a test organism is the A. Minimum inhibitory concentration, serum inhibitory concentration, minimum bactericidal titer, maximum inhibitory titer. The MIC is a basic laboratory measurement of the activity of an antibiotic against an organism. It is the lowest concentration of antibiotic that inhibits visible growth of the organism. It doesn't represent the concentration of antibiotic that is lethal to the organism. So the correct answer is A. Minimum inhibitory concentration. Question number 4. Which of the following clean cat urine culture colony count indicates the patient likely has a urinary tract infection? 10 colony forming unit per ml, 10 to the power 3 colony forming unit per ml, 10 to the power 5 colony forming unit per ml, no gross. Patients with infection often have at least 100,000 bacteria per ml of urine. This means 10 to the power 5 bacteria per ml of urine in the bladder. However, 1/3 of the young women with symptomatic cystitis have less than 100,000 bacteria per ml of urine. The Infectious Disease Society of America definition of cystitis is greater than or equal to 1,000 colony forming unit per ml of a uropathogen. So the correct answer is B. Question number 5. The steam autoclave method of sterilization uses 15 pound per square inch of pressure for 15 minutes, utilizes dry heat for 20 minutes, produces a maximum temperature of 100 degrees centigrade, requires a source of ethylene oxide. The traditional gravity displacement steam sterilization cycle is 121 degrees centigrade for 15 minutes at 15 pound per square inch. So the correct answer is A. Ethylene oxide is an alternative sterilization method. Using autoclave, steam autoclave, you should use temperature 121 degrees centigrade for 15 minutes at 15 pound per square inch. So as we mentioned, correct answer is A. Question number 6. The expected colony count in a suprapubic urine from a healthy individual is 0 colony forming unit per ml, 100 colony forming unit per ml, 1000 colony forming unit per ml, or 100,000 colony forming unit per ml. Urine in the bladder is normally sterile. Suprapubic aspiration removes urine directly from the bladder and yields a specimen free of urethral contamination. So the correct answer is A, because as we mentioned, suprapubic urine is sterile, so no bacteria should be there. So the correct answer is A. Question number 7. An aspirate of a deep wound was plated on a blood agar and incubated aerobically and anaerobically. At 24 hours, there was growth on both plates. This indicates that the organism is an non-fermenter, obligate anaerobes, aerobe, facultative anaerobe. We know that aerobically means in the presence of oxygen. Anaerobically means absence of oxygen. Facultative anaerobes are organisms that can grow under both aerobic and anaerobic condition. So the correct answer is facultative anaerobes, which can grow both aerobically and anaerobically. Question number 8. The proper blood to broth ratio for blood cultures to reduce the antibacterial effect of serum in adult is 1 to 2, 1 to 3, 1 to 10, 1 to 30. Human blood contains substances that may inhibit microbial growth. Diluting blood in culture broth reduces the concentration of these substances as well as any antibiotic that may be present. The recommended blood broth ratio is 1 to 5 to 1 to 10. Dilution less than this may cause the blood to clot, trapping organisms in the clot. Greater dilutions may increase the time to detection. So the correct answer is C, because the appropriate concentration should be range from 1 to 5 to 1 to 10 to reduce the antibacterial effect of serum in adult. Question number 9. A penicillin-resistant Neisseria gonorrhea produces alpha hemolysin, beta lactamase, enterotoxin coagulase. Antimicrobial resistance in Neisseria gonorrhea is widespread. The production of beta lactamase breaks open the beta lactam ring of penicillin, destroying its activity. Thus, Neisseria gonorrhea that produce beta lactamase are resistant to penicillin. So the correct answer is C. B. Question number 10. Which selective media is used for the isolation of gram-positive microorganisms? Colombia CNA with 5% sheep agar, trypticase soya agar with 5% sheep agar, eosine methylene blue, modified tire Martin. Colombia CNA agar contains colistin and nalidic acid, which inhibit most gram-negative organisms, while eosine methylene blue is selective and inhibits gram-positive organisms. Modified tire Martin is selective and inhibits gram-positive organisms and gram-negative bacilli and yeast. So the correct answer is Colombia agar with 5% sheep blood is selective media to isolate gram-positive microorganisms. Question number 11. The most sensitive substrate for detection of beta-lactamase is penicillin, ampicillin, cefoxitin, nitrocephin. The chromogenic cephalosporin test using nitrocephin is the most sensitive and specific test for detection of beta-lactamase. Acidometric tests employing penicillin are less expensive but not as sensitive as the nitrocephin assay. So the correct answer is D, nitrocephin. Question number 12. Staphylococcus aureus isolate has an MIC of 4 microgram per ml to oxacillin. There is uncertainty as whether this represents an oxacillin heteroresistant resistant strain or a hyper producer of beta-lactamase. Strain A susceptible to oxacillin and susceptible to amoxicillin clavulanic acid. Strain B susceptible to oxacillin and resistant to amoxicillin clavulanic acid. Strain C resistant to oxacillin, susceptible to amoxicillin clavulanic acid. Strain D resistant to both oxacillin and amoxicillin clavulanic acid. Based on the above result for oxacillin and amoxicillin clavulanic acid, which strain is heteroresistant? Strain A, strain B, strain C, or strain D? Staphylococcus aureus isolate with an MIC of 4 microgram per ml is resistant to oxacillin as per the clinical and laboratory standards institute CLSI guidelines recommendations. A heteroresistant strain would be defined as a methicillin or oxacillin resistant Staphylo aureus. So MRSA isolate would be reported as resistant to all beta-lactamase agents, beta-lactam inhibitor combination such as amoxicillin clavulanic acid and carbapenems. So the correct answer is D, because Staphylococcus aureus is resistant to both amoxicillin and oxacillin. Question number 13. An enterococcus isolated from multiple blood culture in patient endocarditis should be screened for high-level aminoglycoside resistance, checked for tolerance, assayed for serum antimicrobial activity, tested for beta-lactamase production. Systemic enterococcal infection such as endocarditis are commonly treated with a cell wall active agent, and an aminoglycoside. These agents act synergistically to kill the organism. If the organism is resistant to one or both, there is no synergy and the combination will fail. It is important to detect aminoglycoside and beta-lactam resistance in these cases. Enterococci have intrinsic moderate level resistance to aminoglycosides. Acquired resistance corresponds to very high MIC greater than 500 microgram per ml for gentamicin and is termed high level resistance. So the correct answer is A. Should be screened for high level aminoglycoside resistance. Question number 14. In the Kirby disk diffusion susceptibility test, which variable is critical when testing Pseudomonas species for antibiotic susceptibility to aminoglycosides? Incubation temperature, duration of incubation, cation content of media, depth of agar. Variation in the concentration of divalent cations, primarily calcium and magnesium, affect the results of aminoglycosides, tetracycline, and cholestyl test with the Pseudomonas aeruginosa isolates. A cation concentration that is too high results in smaller zone sizes, and a concentration that is too low increases the zone sizes. So the correct answer is C. Cation content of media, especially calcium and magnesium. Question number 15. The procedure that assures the most accurate detection of MICA-A mediated oxacillin resistance in routine broth microdilution susceptibility testing against Staph aureus is addition of 4% sodium chloride in incubation at 30 degrees centigrade, in incubation for 48 hours, use of cefoxitin for testing. Cefoxitin is used as surrogate for MICA mediated oxacillin resistance in Staph aureus. Staph aureus with cefoxitin MIC more than 4 microgram per ml are considered oxacillin resistance. The Clinical Laboratory Standards Institute CLSI guidelines recommends addition of 2% sodium chloride in incubation at 35 plus minus 2 degrees centigrade and in incubation for 24 hours when performing susceptibility testing of Staph aureus against oxacillin. So the correct answer is D. To use of cefoxitin for testing. Question number 16. When performing a stool culture, a colony type typical of an enteric pathogen is subculture on blood agar plate. The resulting pure culture is screened with several tests to obtain the following result. TSI, acid, put, alkaline slant, no gas, no H2S. Phenylalanine deaminase negative, motility positive, serological typing Shigella subgroup B. The serological typing is verified with new kit and control. The best course of action would be to report the organism as Shigella flexneri without further testing. Verify reactivity of motility medium with positive and negative control. Verify reactivity with TSI slants with positive and negative control for H2S production. Verify reactivity of phenylalanine deaminase with positive and negative control. All of the biochemical and serological reactions listed are consistent with an identification of Shigella flexneri, with the exception of motility. All Shigella are non-motile. So the correct answer is B. Question number 17. Susceptibility testing performed on quality control organism using a new media lot number yielded zone sizes that were too large for all antibiotic tested. The testing was repeated using media from a previously used lot number and all zone sizes were acceptable. Which of the following best explain the unacceptable zone sizes? The antibiotic discs were not stored with the proper desiccant. The depth of the media was too thick. The depth of the media was too thin. The antibiotic discs were not properly applied to the media. All Muller-Hinton agar used for disc diffusion susceptibility testing should be poured to a depth of 4 mm. Depths less than 4 mm, this may associated with excessively large zone and false positive susceptibility result. Agars that more than 4 mm in depth may cause excessively small zone sizes. So the correct answer is C. Question number 18. 3 sets of blood culture were obtained from an adult patient with fever and suspected endocarditis. The aerobic bottle of one set had gross Staphylococcus epidermidis at 5 days of incubation. This indicates that there was low-grade bacteremia. The organism is most likely a contaminant. The patient has line infection. The blood culture bottle are defective. Coagulase negative staphylococci are commonly associated with contaminated blood culture. However, there are also increasing as a cause of true bacteremia. Significant bacteremia in a patient with endocarditis is usually continuous and low grade. In most cases, all blood cultures drawn will yield positive culture. The fact that only one bottle of one set was positive and that the bottle didn't become positive until the day 5 of incubation indicate that this isolate is most likely contaminant. So the correct answer is B. Question number 19. In order to isolate Campylobacter coli jejuni, the fecal specimen should be incubated into selective plating media and incubated in reduced oxygen with added carbon dioxide at 42 degrees centigrade, stored in triptych soya broth before plating to ensure growth of organism, inoculated onto selective plating media and incubated at both 35 degrees centigrade and at room temperature, incubated at 35 degrees centigrade for 2 hours in carry player media before inoculating onto selective plating media. For Campylobacter coli jejuni, require a micro-aerophilic atmosphere for optimal recovery. The use of selective media is recommended for recovery for fecal specimens. Selective media for Campylobacter contains antibiotics to inhibit the growth of enteric gram-negative flora. Unlike other enteric pathogens, Campylobacter coli jejuni grow well at 42 degrees centigrade. So the correct answer is A, because there is reduced oxygen. This means microaerophilic atmosphere, and temperature is 42 degrees centigrade. Question number 20. Sputum is sent to the laboratory for culture from a patient with respiratory distress. The gram stain of the specimen shows many squamous epithelial cells more than 25 per low power field and rare neutrophils. The microscopic appearance of the organism present include moderate gram-positive cocci in chain and diplococci, moderate gram-negative diplococci, moderate placiding gram-positive bacilli, all in moderate amount. This gram stain is most indicative of a pneumococcal pneumonia, an anaerobic infection, and Haemophilus pneumonia or oropharyngeal flora. Sputum specimen quality is assessed to determine if the specimen is representative of the site of infection. The presence of white blood cells is an indicator of infection, and presence of squamous epithelial cells is an indicator of oropharyngeal contamination. In this specimen, there is more than 25 epithelial cells per low power field is an indicator of poor specimen quality, and the bacteria present are representative of oropharyngeal flora. So the correct answer is D. Question number 21. Which type of microscope would be most useful in examining viruses and the structure of microbial cells? Electron microscope, phase contrast microscope, dark field microscope, bright field microscope. Due to the small size of viruses, they are not visible using light microscope. Electron microscope is used to examine viruses and internal structure of microorganisms. So the correct answer is electron microscope. Question number 22. After satisfactory performance of daily disk diffusion, susceptibility quality control is documented. The frequency of quality control can be reduced to twice a week, every other week, every month. Disk diffusion quality control can be converted to weekly testing when 30 days of consecutive testing demonstrates no more than 3 antibiotics per organism combination outside the acceptable limits. So the correct answer is B. Number 23. Disk diffusion susceptibility testing as an antimicrobial agent diffuses away from the disk. The concentration of antibiotic is increased, decreased, unchanged, inoculum dependent. The amount of antibiotic used in disk diffusion susceptibility testing is standardized and constant. Once the disk is placed on the inoculated plate and makes contact with the agar, the antibiotic in the disk begins to diffuse out. As it diffuses into the media, the concentration of antibiotic gets lower the farther it diffuses from the disk. So the correct answer is B. Question number 24. The most common causes for failure of gas bag anaerobic jar to establish an adequate environment for anaerobic incubation is the failure of the oxidation reduction potential indicator system, due to the deterioration of methylene blue, the failure of the packet to generate adequate hydrogen and or carbon dioxide, condensation of water on the inner surface of the jar, catalysts that have become inactivated after repeated use. The most two common causes of failure of the gas bag system is a defective gasket in the jar lid that allows escape of gas from inside the jar and inactivated catalyst pellet. So the correct answer is D. Question number 25. Which of the following is the most appropriate method for collecting your specimen from patient with an indwelling catheter? Remove the catheter, cut the tip, and submit it for culture. Disconnect the catheter from the bag and collect urine from the terminal end of the catheter. Collect urine directly from the bag. Aspirate the urine aseptically from the catheter tubing. Indwelling catheters are closed system and should not be disconnected for specimen collection. Urine sample should not be collected from the catheter bags, and fully catheter tips are unsuitable for culture because they are contaminated with colonizing organisms. Urine for indwelling catheter should be collected by aseptically puncture the tubing collection board. So the correct answer is D. Aspirate urine aseptically from the catheter tubing. These are the 25 questions in microbiology. Please like, share, and subscribe to receive the new video for the scientist and technologist license exam. Wassalamu alaikum wa rahmatullahi wa barakatuh.